Researchers from University Medical Center UMC Utrecht, Netherlands, for the first time successfully combined volumetric bioprinting with multi-electrowriting. This combines the speed and cell-friendliness of volumetric printing with the structural strength needed to create functional blood vessels. The study by the Biofabrication Lab of Regenerative Medicine Center Utrecht RMCU was published in the journal Advanced Materials. Volumetric printing is a technique that was pioneered for bioprinting by the RMCU Biofabrication Lab in the year 2019. It is a fast technique which allows cells to survive the printing process. However, because this type of printing is done in cell-friendly gels, the resulting prints are structurally not very sound. This is a problem for blood vessels which have to be able to withstand high pressures and bending. For this reason, a merger of volumetric bioprinting and multi-electrowriting was pursued. Multi-electrowriting is a highly accurate type of 3D printing that works by directing a narrow filament of molten biodegradable plastic. It is able to produce intricate scaffolds that are mechanically strong and able to deal with force. The downside here is that they can't be printed with cells in there directly because of the high temperatures involved. Therefore, volumetric bioprinting was used here to solidify cell-laden gels onto the scaffolds. How to merge electrowriting with volumetric printing? The process starts with the creation of a tubular scaffold using multi-electrowriting. This is then submerged into a vial with photoactive gel and placed in the volumetric bioprinter. In principle, the laser of the printer can selectively solidify the gel that sits on and around the scaffold. The researcher says that in order to get this right, they had to place the scaffold exactly center in the vial. Any deviation from the center would mean that the volumetric print would be offset. But they managed to center it perfectly by printing the scaffold on a mandrel that they fitted to the vial. In this study, the researchers tested various thicknesses of the scaffold, which resulted in more or less strong tubes. Finally, they also tested various placements of the bioprinted gels. They could either be placed on the inner side of the scaffold, inside the scaffold itself, or on the outside of it. By using two differently labeled stem cells, the team was able to print a proof of principle blood vessel with two layers of stem cells and seeded epithelial cells in the center to cover the lumen of the vessel. The design could also allow for holes in the side of the print, giving the possibility for controlled permeability of the vessel for the blood to do its function. Finally, the researchers also created more complex structures like forked vessels and even vessels with venous valves that were functional in maintaining a unidirectional flow. The researcher says that this was a proof of principle study. What they now need to do is replace the stem cells with functional cells that are part of a real blood vessel. That means adding muscle cells and fibrous tissue around the epithelial cells. Their goal now is to print a functional blood vessel.